Welcome to the first video of MYP Masterclass for Biology. Today we will be learning about microscopy, which is a topic that is usually taught during MYP 4. Microscopy is the use of microscopes to magnify objects that are too small to be seen by the naked eye. Microscopy uses a microscope to produce a magnified image and separate the details of this image to make it visible to the naked eye. There are two main types of microscopes: a light microscope and an electron microscope. A light microscope uses visible light to magnify biological samples with the highest magnification of between 1000 and 1500. A light microscope was the first microscope to be created during the early 1600s by Zacharias Janssen, and it was used to make multiple discoveries in biology, such as the existence of bacteria. On the other hand, an electron microscope has a much higher magnification of over 100,000, and it is used to observe much smaller samples such as viruses. For this video, we will be exploring the light microscope and its structure and components. Here is a diagram of a light microscope that we can now label. The first component is the eyepiece or ocular lens, which is where the user looks through to see the sample, and the diopter adjustments helps them adjust them to fit. The arm contributes to the structure and transport of the microscope. It is important that when transporting a microscope, you grab it by the arm and base to ensure that it remains intact. Below this are the adjustment knobs, a coarse adjustment knob and a fine adjustment knob. The coarse adjustment knob makes more significant changes while focusing the sample, while the fine adjustment knob makes slight changes. Near the bottom of this microscope is the on and off switch to turn the microscope on and off. And some microscopes may have an element to adjust the intensity of the light as well. Then there's the base as well as the light source. Higher up, we can find the main part of the microscope, which consists of the diaphragm, condenser, stage clips or slide holders, and the objective lens and revolving nose piece. The diaphragm and condenser direct the light from the light source to the sample, which is placed on the stage and secured with stage clips or slide holders. Finally, the magnification of the sample can be changed based on the objective lens that is in use. Although it may vary from microscope to microscope, most will have a 4x, 10x, and 100x objective lens, which can be changed using the revolving nose piece. Now that we know the structure and components of the microscope, we can discuss two important terms: magnification and resolution. It is very important to know how to distinguish these two terms. Magnification is the number of times an image is enlarged. So for example, if the magnification is 10x, it means the image has been enlarged 10 times. On the other hand, resolution refers to the ability of a microscope to distinguish the details in a sample. For now, this is all you will need to know, but in the future you may have to do some calculations using these values. Thank you for your attention.